After fertilization, the fertilized ovum undergoes segmentation, and when it will reach 16 cell stage, we generally call it morula. Morula, outer side of the morula is covered by which prevents implantation, zona pellucida. Now, when zona pellucida will be removed, then it will gradually enter into the uterine wall, right? Now, some fluid from the uterine cavity will enter into the morula, and one fluid-filled cavity will be formed like this. That is called blastocyst. Now, the cells of the morula will rearrange themselves. Some cells lying in groups, and some cells go to the periphery. They will form trophoblast. So after rearranging the cells which lie around the periphery, we call it trophoblast. And groups of cells which lies within the cavity, that is called inner cell mass or embryoblast. From this cell, embryo will develop. That's why it is called embryoblast. And peripheral cells, trophoblast, that is extraembryonic cell, they are not involved in the formation of embryo. Now these embryoblast cells will again, they will rearrange themselves into two layers. When cells become differentiated from other, we call it differentiation. When one cell becomes differentiated from other, then we call it differentiation. So here the cells have been differentiated. The cells which lying, these cells, columnar cells, we call it epiblast. These columnar cells is called epiblast. And the flattened cells which lies facing the cavity, that is called hypoblast. Hypoblast and epiblast, right? Now, some cells from the hypoblast, they will spread around this cavity. These cells are coming from hypoblast and they are lining this cavity that is known as Husser membrane. These cells called now Husser membrane. Husser membrane. Husser membrane. And this cavity is now known as primary yolk sac. Now this cavity is known as primary yolk sac. Primary yolk sac. Right. Now, a cavity appears between epiblast and trophoblast. Here lies trophoblast. Now, a cavity appears between trophoblast and epiblast.
Accepted.
Yeah. Now this cavity is known as amniotic cavity. The cavity which appears above the epiblast cell and the trophoblast, this cavity is known as amniotic cavity. Now, this amniotic cavity is lined by, its roof is lined by cells which are derived from trophoblast. here we have seen this primary yolk sac cavity is lined by cells coming from the hypoblast. But here this cavity is lined by cells which is coming from the trophoblast. Then it is derived from trophoblast. Now this cavity is known as amniotic cavity, and this is the primary yolk sac. So now our embryo is at the two cell stage, epiblast and hypoblast. Is it clear? Now, third event. Now from trophoblast cell will form another layer that is called extraembryonic mesoderm. From trophoblast cell will give rise to extraembryonic mesoderm. Thus, extraembryonic mesoderm will lie between usual membrane and trophoblast. Actually, it is trophoblast. Now here you see this cell is the trophoblast. The red one is mesoderm. The red one is extraembryonic mesoderm. So this extraembryonic mesoderm actually has been given rise to by trophoblast. Now here you see this is called after formation of the extraembryonic mesoderm, this primary yolk sac will now be converted as secondary yolk sac. Sorry. Now it is secondary yolk sac. Secondary yolk sac. Secondary yolk sac. Actually, এখানে তোর ছবিটা আঁকতে আমার অসুবিধা হচ্ছে, বুঝলি তো? একটা আঁকতে একটা বুঝে দিচ্ছে, যখন when secondary yolks will develop, primary this mesoderm will develop. Actually, due to the proliferation of the mesoderm, that cavity will be small in size. যদি এখানে ধর আরেকটা লেয়ার ডেভেলপ করে, if one layer develops, then it will automatically be reduced in size. Then it is known as secondary yolk sac. Now, pro structure আমি আগে বলছি, the most peripheral one is the trophoblast. Deep to trophoblast, the red one is the extraembryonic mesoderm, and deep to that, around the yolk sac, this is called Husser membrane, and around the roof of the amniotic cavity, that is the amniogenic cell. It is amniogenic cell. It is a Husser membrane. Amniogenic cell is derived from the yolk trophoblast, and this Husser membrane, the cell is coming from the hypoblast. It is clear? Backbench. Is it clear? These three layers, most outer layer is trophoblast. Red one is extraembryonic mesoderm. Inner cell, there are two cavity. Upper cavity is known as amniotic cavity. Lower cavity is known as yolk sac. Yolk sac is lined by Husser membrane. Husser membrane cell has come from the hypoblast, and the amniogenic cavity by amniotic cavity, its roof lined by cells which has come from the trophoblast, and its floor is formed by the epiblast. Right? এবার আমি এটাকে সব মুছে দিয়ে আবার নতুন করে আবার একটা আঁকছি। Here you see, this black one is the primary mesoderm, which layer has given rise to trophoblast. Here you see one cavity has appeared within the primary mesoderm. This cavity is known as extraembryonic coelom. This cavity is known as extraembryonic coelom. Here you see that extraembryonic coelom has not spread here, just above the amniotic cavity. This coelom has not reached here. This part will later on form connecting stalk. Here will develop placenta. This is the connecting stalk. Placenta, umbilical cord, etc. will develop here. So, up to that level, this coelom, this cavity has not reached. So, this part is known as connecting stalk. Right. When this germ disc will appear, then we call it, it has got two pole, embryonic pole, embryonic pole. This is called embryonic pole, where this two cell layer has developed, that is called embryonic pole, and opposite pole is embryonic pole. Right. Now, next event. We know, তোদের তো এগুলো তো আগে একবার হয়ে গেছে তো, ফার্স্ট উইক ডেভেলপমেন্ট, সেকেন্ড উইক ডেভেলপমেন্ট, থার্ড উইক ডেভেলপমেন্ট তো হয়ে গেছে। আমি তো জাস্ট ওইটুকু বলবো যেটুকু আমার রিলেটেড টু কার্ডিওভাস্কুলার সিস্টেম। নাও, হ্যাঁ কি আরে বই তো পড়তে হবে, বই না বলে বুঝবি কি বইটা পড়তে হবে তোকে আগে। নাও let us see here. Now we know that during third week the germ layer becomes 3-layered. During second week germ layer becomes 2-layered. The 2-layered, here everything during the second week only week of 2. Here you will find 2 cavity. During the second week you will get everything 2. Number 1, 2 cavity, that is yolk sac and amniotic cavity. 1. This embryonic disc is 2-layered, epiblast and hypoblast. Then extraembryonic mesoderm, this extraembryonic mesoderm, here appear cavity, then it has become extraembryonic coelom, and after the appearance of the cavity, this extraembryonic coelom has been divided into two parts. One part is lining the trophoblast, and one part is lining the Husserl membrane or yolk sac cavity. Which one is covering the yolk sac cavity? That extraembryonic mesoderm is known as splanchnopluric. It is known as splanchnopluric splanchnopluric extraembryonic mesoderm, and or visceral extraembryonic mesoderm, and which is lining the trophoblast, that is known as somatopluric. Somatopluric extraembryonic mesoderm or parietal mesoderm. This is called visceral mesoderm, which is lining the yolk cavity and which is lining the trophoblast, is known as somatopluric extraembryonic mesoderm or parietal mesoderm. Now, তাহলে এখানে দুটো হলো। Now two membrane. Here it gives rise to two membrane. One is amniotic membrane and one is, what is the name of other membrane? Amnion and chorion. There are two things, amnion and chorion. What is called chorion? This layer, these two layer, trophoblast and extraembryonic mesoderm, these two layer will form chorion. এটা বলে chorion membrane, chorion. Right? And here you see this part. Here you see the amnion. This layer is known as amnion. Amnion means, amnion is the, actually here it is the connecting stalk, right? Now just below the connecting stalk, here there is one cell layer. I could not draw it. That cell layer is amniogenic layer. এখানে একটা লেয়ার ছিল amniogenic layer. Amniogenic layer. To start with, I told you that this amniogenic layer is derived from the trophoblast, which has formed the roof. That layer and this embryonic mesoderm will form the amnion. This one will form the amnion. Clear হলো না এটা? Clear হয়েছে? হয়নি? amnion and chorion. amnion and chorionটা তো ক্লিয়ার হয়নি, chorionটা ক্লিয়ার হয়েছে, amnion ক্লিয়ার হয়নি, তাই না? amnion is the, I have already told you that the extraembryonic mesoderm has not actually completely separated the roof of the roof of the amniotic cavity. তাহলে the roofটা কিছুটা রুফটা কমে কিন্তু ম্যাক্সিমাম রুফ হ্যাজ বিন কভার্ড। ছবিতে আমি যদি দেখাতে পারিনি, ধর এখানে যে রুফটা আছে, দ্যাট রুফ, এখানে যে রুফটা আছে, এটা তাহলে কী কী সেল আছে? অ্যামনিওজেনিক সেল প্লাস আমি যেটা বলতে চাইছি যে দের আর টু লেয়ার, একটা হচ্ছে স্প্ল্যাঙ্কনোপ্লুরিক লেয়ার, একটা সোম্যাটোপ্লুরিক লেয়ার। এইখানে কি লেয়ার থাকবে, সোম্যাটোপ্লুরিক না স্প্ল্যাঙ্কনোপ্লুরিক? এই পর্যন্ত হচ্ছে তোর এই পর্যন্ত, এটা হচ্ছে স্প্ল্যাঙ্কনোপ্লুরিক। শুধু মনে রাখ যে স্প্ল্যাঙ্কনোপ্লুরিক লেয়ার and the cell forming the roof, these two will form the amnion. Somatopleuric layer and the trophoblast যে লেয়ারটা আছে এখানে, তার আগে আছে হিউসার মেমব্রেন অ্যান্ড তোর কি বলে somatopleuric layer will form the chorion. বুঝতে পারলি? Now third week development. During third week, what important events take place? During third week, the two cell layer of germ disc will form three cell layer, and the most important event which takes place during the third week, যদি তোদের থার্ড উইক একটা আসে এরকমভাবে, থার্ড উইক ডেভেলপমেন্ট, the most important event during the third week is first হচ্ছে appearance of primitive streak, থার্ড উইক 7/7/14 ডেতে অ্যাপিয়ার হয়। তোদের তো একটা এমসিকিউ ছিল, একটা এমসিকিউ এসেছিল এবার, primitive streak develops on what day? 15 days. The primitive streak, if you count the word primitive streak, 15 word the primitive streak হয়। primitive streak গুনে দেখ 15, তো 15 days it appears. So the most important event during the third week is the appearance of the primitive streak. Then another event that is called gastrulation. What is gastrulation? Gastrulation is the process by which two germ cell layer become three germ layer. এই প্রসেসটাকে বলে gastrulation. Third important event is development of notochord. তোদের এসেছিল development of notochord. Fourth হচ্ছে neurulation. Neural plate will develop, neuroectoderm will develop, neurulation. So these are the third week development. And another important development during the third week, another mesoderm will appear, that is called secondary mesoderm. এখানে প্রাইমারি mesoderm already told you, প্রাইমারি mesoderm তো দেখলি, এবার হচ্ছে secondary mesoderm. When secondary mesoderm will develop in between two layers, then it becomes three-layered germ disc. Three-layered germ disc means ectoderm, mesoderm and endoderm. This is called three germ layer. When it appears during the third week of intrauterine life. Now, how it appears? Here I will draw one picture, that is I am only drawing the embryonic disc area. This is the During the last part, this is called hypoblast, this is called epiblast. During the last part of second week, some cells here will be columnar in shape. It is more or less flat or cuboidal in shape. Some cells become columnar. This area we call it procaudal plate. Procaudal area. Procaudal area. Procaudal area. During the last part of second week. First, during the second week, the germ embryonic area but germ is like circular. It becomes circular. Suppose here this area has developed. This area here it is developed. This is called procaudal area. Procaudal area. With the appearance of the procaudal area, what will happen? The axis will be determined. With the appearance of the procaudal layer, axis will be determined. This is the cephalic axis, cephalic side. This is the caudal side. This is the cephalic side. This is the caudal side. With the appearance of the procaudal area, what is procaudal area? When some cells of the hypoblast become columnar, that part is known as procaudal part. With the appearance of the procaudal area, axis will be determined. That is called caudal cephalic axis. This is called caudal axis, right? And as hypoblast lies below, so dorsoventral axis will be also developed. It is the dorsal side, it is the ventral side. Dorsoventral axis will develop. If there is dorsoventral and cephalocaudal, so right and left axis will also develop. Three axes we can now determine with the appearance of the procaudal plate. Procaudal plate key? It is nothing but some cells, some flattened cells or cuboidal cells of the. समझ में नहीं आ रहा है ना? You can't understand, can't follow. If you can't follow, you will stand up and tell me, sir, here I am feeling difficulty. Otherwise, you will remain unclear throughout your life. তোদের প্রত্যেকের খাতা ধরে ধরে ডেকে ডেকে আমি দেখাবো আর কি তোরা কোথায় কী ভুল করেছিস, কোথায় তোদের গবেষণালব্ধ, হ্যাঁ অনেকেই গবেষণালব্ধ জিনিস লিখেছিস, হ্যাঁ মানে বোধহয় সেটা নেক্সট জেনারেশন আসবে হয়তো, আমরা এখন জানি না, সেরকম জিনিসও লিখেছিস আর কি, হ্যাঁ সেগুলো সব দেখিয়ে বলবো আর কি, খাতাটা দেখাও। So now from circular area, it will now with the appearance of the primitive streak will now develop on the primitive streak develops where? In the region of epiblast or hypoblast? In the region of epiblast. Some epiblast cells, here you see, at the beginning of the third week, when the primitive streak appears, some cells at the caudal aspect, it is the caudal aspect, it is the cephalic aspect, কারণ এটা the procaudal plate, some cells proliferate, and that proliferated cells is the appearance of the primitive streak. With the appearance of the primitive streak, the circular germ disc will appear to some extent oval shaped. এবার oval shaped হয়ে যাবে। It is oval shaped. Here lies the procaudal plate and here lies the germ disc. কি বলে? Primitive streak. primitive streak. Primitive streak lies on the epiblast side and procaudal actually lies on the hypoblast. Hypoblast cell proliferate kore, it has given rise to procaudal plate. And epiblast proliferate to form primitive streak. It becomes linear. Some cells become more proliferated, so it becomes like this. It is called primitive node. It is called primitive streak. Primitive streak. It is a primitive node. Some depression appears here, that is called primitive pit. Primitive pit. How intraembryonic slope develops, it is very important. The epiblast cells, give attention to, epiblast cells will migrate through, epiblast cell will migrate through primitive streak. So actually primitive streak, from the primitive streak, the cells will pass through the primitive streak. They will form two kinds of cells. One kind of cells will displace the, or will replace the hypoblast cells, and that will be the definitive endoderm. এটা ছিল আমরা hypoblastটাকে আমরা in some books it is written hypoblast is primitive endoderm and epiblast is primitive ectoderm. Now definitive endoderm, when it will happen? When the epiblast cells will pass through the primitive streak and will replace this hypoblast cells, and it will form the definitive endoderm. In between two cells, from this primitive endoderm, some cells will enter and it will come between the two cells, between the epiblast and hypoblast. That will form the secondary mesoderm, and the remaining cells of the epiblast will form the definitive ectoderm. So in one word, this one will remain throughout our life, that from epiblast every layer means 3 germ layer develops. Epiblast will give rise to this 3 germ layer: ectoderm, endoderm and mesoderm. If you are asked ectoderm and endoderm and mesoderm, from which these 3 layers have come, they have come from epiblast. From epiblast. So epiblast will give rise to all the 3 germ layer. And from these 3 germ layer, all our body parts will develop. Everything, every organ, every system will develop from 3 germ layer. It clear too? Now, here you see, I am This is the secondary mesoderm, right? Now, this secondary mesoderm will be divided into stand up. You. Bolo. Bro, secondary mesoderm comes from where? Secondary mesoderm comes from where? Which actually, who is producing this secondary mesoderm? बॉडी का जो है ना सब पार्ट तो यहाँ से डेवलप होगा ना, तो हाउ विल यू नो? কুড়ির বেশি তো কেউ নাম্বার পায়নি, তোদের এরকম যদি হালাত হয় তাহলে তো বুঝতেই পারছিস, ফেল করা মানে একটা পুরো ইয়ার লস। সেকেন্ড ইয়ারটা যদি না পাস করতে পারিস, ইফ ইউ ক্যানট পাস দ্য অ্যানাটমি, ফিজিওলজি অ্যান্ড biochemistry, there is a chance of your loss. In case of other, if you fail, you will be promoted to next class and you have to clear later on. But here, until you clear these three subjects, you will not be promoted to next. You have seen that when primary mesoderm developed, some cavity will appear, and after the appearance of the cavity, this primary mesoderm again is divided into two, somatopluric and splanchnopluric. Here also cavity will appear. Cavity will appear within the secondary mesoderm. Cavity will appear within the secondary mesoderm. Suppose this is the cavity. Now, what will be the name of this cavity? There we call it extraembryonic coelom. Here we will call it intraembryonic coelom. With the appearance of the intraembryonic coelom, now some part of the mesoderm lies in contact with the endoderm, and some part is lying in contact with the ectoderm. This is ectoderm. Primitive streaker dikta ectoderm. Eta endoderm. Eto. Now this mesoderm, interembryonic mesoderm, which is in contact with ectoderm, that is called somatopluric mesoderm. And this one, which is facing the yolk sac, এখানে তো yolk sac আছে, secondary yolk sac আছে তো নিচে, that is called splanchnopluric mesoderm, splachnopluric mesoderm, right? Now, from this splachnopluric mesoderm, our heart will develop. এতটা দূর আসার কারণ হচ্ছে এটাই, হার্টটা যে স্টার্ট করবো, হঠাৎ করে হার্ট স্টার্ট করে দিলাম, কোথা থেকে হার্টটা স্টার্ট হলো বুঝতে পারবি না, তাই এতটা জার্নি। এবার বুঝতে পারলি? তাহলে যদি বলে ফ্রম হোয়ার হার্ট উইল ডেভেলপ ফ্রম হুইচ পার্ট অফ দ্য ইন্ট্রাএমব্রিয়নিক মেসোডার্ম, heart it's a mesodermal in origin, heart and blood vessels, so mesodermal origin. But from which mesoderm? Splachnopluric mesoderm. Now as heart is developing, so this is known as cardiogenic area. Now this intraembryonic cilium, actually that intraembryonic আমি তো একটা ছোট্ট পার্টে গেছি, it is a big part, পুরো এটা নিয়ে থাকবে। From that intraembryonic cilium, our body cavity, our peritoneal cavity, our pleural cavity, all will develop from the cilium. From this cilium, from pleural cavity, pericardial cavity, and peritoneal cavity, all will develop from this cilium, intraembryonic cilium. Now, suppose এটা হচ্ছে তোর, I am viewing it from above downwards. That means that is the epiblast বা কি বলে epiderm, আর তার নিচে আছে মেসোডার্ম, ইন্ট্রাএমব্রিয়নিক মেসোডার্ম, তার নিচে আছে এন্ডোডার্ম। এখন ধর অ্যাপিয়ার হয়েছে তোর সেই ক্যাভিটিটা, দ্যাট ক্যাভিটি, এখানে আছে প্রোকডাল প্লেট, এখানে আছে প্রিমিটিভ স্ট্রিক, এখানে ডেভেলপ করে তো নোটোকর্ড। When this cilium will develop, that cilium actually will not cross the midline. সে সিলমটা এরকমভাবে সাইড দিয়ে যাবে আর কি। It is the intraembryonic cilium. It is lying between the two layers of the ectoderm and endoderm মাঝে mesoderm, সেই mesoderm মধ্যে এই ক্যাভিটি অ্যাপিয়ার করেছে। মিডলাইন পার্ট, এই সিলোমের যেটা কেফালিক টু দ্য প্রোকোডাল প্লেট, এই পার্টটা উই কল ইট পেরিকার্ডিয়াল স্যাক। ওইটা হচ্ছে পেরিকার্ডিয়াল স্যাক। এবার একটু মনে মনে কল্পনা করো, এখানে পেরিকার্ডিয়াল স্যাক, ওই পার্টটুকু, একটু পেরিকার্ডিয়াল স্যাক, তারপরে এখানে তো ডেভেলপ করে পেরিটোনিয়াল স্যাক, কী বলে তোর প্লুরাল স্যাক, নিচে হয়তো তোর পেরিটোনিয়াল স্যাক। ফ্রম হিয়ার ইট উইল ডেভেলপ পেরিকার্ডিয়াম। সো if this part is pericardium, this gap is pericardium, so its roof is covered by which mesoderm, and its floor is covered by which mesoderm? इंट्राएम्ब्रियोनिक मेसोडर्म का अंदर, इंट्राएम्ब्रियोनिक जो मेसोडर्म है इसका अंदर एक कैविटी अपीयर किया, दैट कैविटी इज इंट्राएम्ब्रियोनिक सीलम, दैट इंट्राएम्ब्रियोनिक सीलम व्हिच लाइज just cephalic to the procaudal plate, that part is known as pericardial sac. So pericardial sac has split the intraembryonic mesoderm into two. Some mesoderm lies towards the ectoderm, and some are facing the endoderm. So which mesoderm, intraembryonic mesoderm lies on the roof of the pericardial plate, the pericardial sac, is the which pericardium? Somatopleuric, very good. And which lies on the floor? Splanchnopluric, very good. বুঝেছে এরা বুঝেছে, বুঝতে পেরেছিস? देखो क्लियर जब तक नहीं होगा ये हमारा एक्टोडर्म होगा, ये हमारा एंडोडर्म। इसका बीच में डेवलप किया हमारा मेसोडर्म, इंट्रा एम्ब्रियोनिक, वो तो एम्ब्रियोनिक एरिया है ना, तो इंट्रा एम्ब्रियोनिक। तो इसका अंदर सपोज यहाँ पे हमारा एक कैविटी अपीयर किया, कैविटी अपीयर करने से जो ये मेसोडर्म जो है यहाँ पे दो भाग हो गया, एक भाग इधर चला गया, वो इधर चला गया। इस भाग का जो है नाम है क्या, और इस भाग का क्या नाम है? ये है स्प्लैंक्नोप्लूरिक और ये है सोमेटोप्लूरिक। सो इट नीचे है ये फ्लोर, इधर रूफ, राइट? তাহলে মোটামুটি বুঝে গেছি, আর ভয় নেই। নাও from this pleural cavity, হ্যাঁ কি বলে, pericardial sac, pericardium will develop. Now there will be head fold of the embryo. Do you know the head fold of the embryo? ये पूरा जो है ना, ये ऐसे घूम जाएगा। There will be head fold of the embryo. जब head fold of the embryo हो जाएगा, then the pericardium which was roof will be floor and floor will be roof. এটা ধর আছে তো, ধর এখানে আছে, ধর এখানে তো বইয়ের এখানে ধর আছে, এটা জিনিসটা আছে। এবার আমি যদি পুরো মিটার ঘুরিয়ে দিই, একদম পুরো ঘুরিয়ে গেলো, হেড ফোল্ড হয়ে গেলো আর কি। যখন হেড ফোল্ড হয়ে যাবে না, তখন পুরো জিনিসটাই ঘুরে যাবে, ওটাও ঘুরে যাবে। মাথাটা তখন ঘুরে, মাথা এরকম ছিল তো, এরকম ছিল, এবার এরকম হয়ে গেলো, এরকম ছিল জিনিসটা, ধর এখানে লেগেছিল একটা এবার এমনি যেমন করলাম, তখন কী হলো? তাহলে এটা উপরে চলে গেলো, এটা নিচে চলে গেলো, পুরো ঘুমিয়ে গেল। রুফ হয়ে যাবে ফ্লোর, হয়ে যাবে রুফ। When there will be head fold, the splanchnopluric mesoderm, which was on the floor of the pericardial sac, now it will be found on the roof of the pericardial sac. Suppose this is pericardial sac. Now I am drawing it after head fold. It is after head fold. After head fold. This is splanchnopluric mesoderm. Splanchnopluric mesoderm. And this is somatopleuric mesoderm. This is the roof. It is a roof. It is a floor. Now here will develop, this is the mesoderm, splanchnopluric mesoderm. Here will develop two tube, two endothelial tube will develop. Two endothelial tube will develop like this. Now they will invaginate. They will, from above downwards, it will invaginate the pericardial sac, and pericardial sac will gradually cover it, and that pericardial sac will ultimately form the pericardium. Now that much will keep it in your memory, that is, from the splanchnopluric mesoderm, heart tube develops. After head fold, where it lies on the roof of the pericardial cavity. That is the main thing of today's discussion. That much